So as she mentioned, I'm in computer science, working under Professor Satish Narayanasamy. And the last thing I wanted to add is that I'll be graduating soon and on the job market, looking to go into industry. So if anybody happens to know people hiring in bioinformatics, I'd be happy to talk after the talk. Okay, so today presentation is on accurately benchmarking phased variant calls. And today I'd like to talk about two different problems that I've worked on over the last year or so. But before I get into that, I'll start off with a bit of background on the problem that I'm working on, which is whole genome sequencing evaluation. As you all know, sequencing cost is rapidly declining over the past few decades, and the cost per human genome went from over $10 million for the first effort. Now it's well under 1,000. And with that lowering of cost has come an exponential growth in the number of genomes that are being sequenced. And then some of the downstream applications are, of course, genome-wide association studies, drug development, clinical diagnostics, and benchmarking new methods and technologies. And for all of these downstream applications, once you have sequenced a genome, it's important to be able to compare the genomes of different individuals. And so the way that this is typically done, because genomes are mostly identical, around 99% similarity across individuals, typically they store everything in a different spaced format. So you have the reference human genome along with just storing the specific locations that there are any mutations, such as a G to C here, a deletion of a T, an insertion of an A, and so forth. And this difference-based results are typically stored in variant call format, where you note the position of the mutation, and then what the reference genome said, and then what the alternate sequence is. So here we can see at position 4, see, here we go, at position 4, a G was mutated to a C, and so forth. Now, what I'm going to be focusing on is specifically benchmarking the results of these variant calls. And the motivation for that is that there's been a ton of new sequencing technologies that have emerged over the last few years. And along with those new technologies have come a lot of different bioinformatics pipelines, each with their own advantages and disadvantages. Now, a simple example of how these technologies are benchmarked. As I said before, you start off with your reference and then the sequence that is reported by your sequencing, and you only store the differences. So technology 1 might say that at position 3, a C becomes an A, and then later on a T becomes an A as a mutation, whereas a second technology might slightly differ. They agree with the T becoming an A at position 6, but instead it says later at position 9 an A becomes a G. So something that's important to do is to know, is technology 1 more accurate? Is technology 2 more accurate? And to understand the pros and cons of the two different technologies. In order to do that, typically you have some form of ground truth. And this ground truth is typically created using multiple different sequencing technologies and sequencing DNA at much higher coverage, so that you're more confident in the set of calls that you have on this data set. So here the ground truth might agree with the T becoming an A, and then agree with the second technology with the A becoming a G at position 9. And so using this ground truth, you can then say whether your variant calls were correct or false positives from your different technologies and get a good understanding of how accurate they each are. Now, when people do this analysis, they typically stratify by the different types of variants because some are more likely than others, and they have different characteristics. So SNPs, or single nucleotide polymorphisms, are when there's a substitution of one base to another. Indels are insertions or deletions less than 50 bases in size, and structural variants are insertions and deletions that are larger than 50 base pairs. And SNPs are the single most common mutation but insertions and deletions account for a large fraction of the total bases that differ. Now, today I'm going to start off by focusing just on small variants, because typically they're evaluated separately from structural variants, because there historically were different sequencing pipelines in order to evaluate the two. So for now, we will just look at the small variants, which are SNPs and indels. And typically the performance is evaluated in the form of a precision recall curve. So on the x-axis, you have recall, and on the y-axis, you have precision. So precision is basically out of all the total calls that you made, how many of those were correct? Whereas recall is out of all the variants that did exist, how many did you correctly identify? So obviously, we want to correctly identify all the variants that do exist, and then we want everything that we do call to be correct. So ideally, you want your precision recall curve to go to the top right hand of this graph. And here we can see the precision recall curves for two different example sequencing technologies or pipelines for indel variants. Now, this is all well and good, but there is a problem once you start talking about complex variants. So when we categorized into SNPs and indels and structural variants, complex variants are any type of variant that cannot be represented by a single mutation. So it's not just a single mutation of one base or a single insertion. It's when there's lots of variants that kind of happen nearby one another, and it's really hard to tell what actually happened. So here is an example of something that's not quite a complex variant, but it does illustrate the problem that complex variants cause. So here, if you have your reference sequence, and then two different query sequences, so say you sequence the same individual using two different technologies. For query one, you've represented the variation as a deletion here at this position, a deletion of a T, and then an insertion of an A a little bit later. Whereas in query number two, you say that this T has mutated to an A. And if you note, the actual resulting sequences are the exact same. There's one A, and then 5 T's, and then 3 A's. So between query 1 and 2, your sequences are the exact same. So your sequencing technologies have identified the exact same underlying DNA sequence, but whether you represent this change as an insertion and a deletion versus a mutation will then impact your downstream benchmarking results. So here is an example kind of illustrating this problem, where we have two different pipelines that actually, so this is real data for whole genome sequencing, where we used two different kind of types of variant representation. So in yellow, it's the variant representation that favors insertions and deletions. And then in purple, the variant representation that favors SNPs or single base mutations. And even though the reported sequence is exactly the same, because of the variant representation, one pipeline seems to do much better on indel precision, but worse on SNP recall. So that's kind of the motivation for why variant representation matters in the area of genome sequencing evaluation. So an example, kind of instead of at a high level, more at a concrete level, is if you had a ground truth that used one representation for a complex variant that uses substitutions versus a representation that prefers insertions of deletions. Again, the actual sequences are the exact same, both the reference sequence and the truth. However, it's just the variant representation that has changed. And now when you shift towards evaluating two different technologies that have different sets of variant calls, the representation that you chose for your ground truth will end up affecting what decisions you make in comparing the technologies. So for example, if we were to use the first representation that favors substitutions, then technology one will seem to do much better because it has two substitutions correctly called and no indels. Whereas if we instead used the second representation that prefers indels, then we see that technology number two gets one true positive variant call, whereas the first technology seems to have not made any correct calls. So the variant representation really impacts your results. And the solution to this problem comes in the form of variant normalization. So you may have heard variant normalization is standard practice. However, there's typical variant normalization and then some extensions that we add on to it. So for example, if you have a reference and query sequence that differ, you would align them to one another and represent the variance in some form. So this form right here shows two bases changing to two other bases. Then over here, AATC becomes AAATC. Now, the standard process would be to first decompose any substitutions. So over here, this AG to TC becomes two separate substitutions. And the next step is for your insertions and deletions to be trimmed. Because over here, the bases T and C match exactly. So we can trim that off the end and we can trim the A off the beginning to simplify it to only include the difference plus just one preceding base. And the last step that's typically done is left shifting. So here we have a single A being added, and you can actually add that A anywhere along that sequence of A's. And so it's standard practice to insert it at the leftmost position. So over here, we've shifted the insertion from position 7 to position 4. And this is kind of the typical process of variant normalization. And this is sufficient to ensure that for any individual variant, it has a unique representation. And the problem comes once you start working with complex variants. So if you have these multiple variants that are all nearby one another, then suddenly there's multiple different ways to represent this change. So here's another example here where the reference and query sequences are the exact same, and you're only changing the alignments and therefore the variant representations. So instead of using two substitutions and one insertion, you could also use one deletion and one insertion to represent the same variation. So the problem that we're trying to solve is how do you choose between these two representations and decide which one is better? So what we looked at is something called alignment-based normalization. So back here, the two representations result from two different alignments. So if you could standardize the parameters for the alignment, then you could have standardized representations for these more complex variants. So alignment is typically done with something known as an affine gap penalty, which is basically you're trying to model the likelihoods of substitutions versus insertions and deletions. And so the simplest way to penalize an insertion or deletion would just be directly proportional to the length of that insertion or deletion. And that would be a simple linear gap penalty. But what's typically done is an affine gap penalty, where you do have a linear increase in terms of the length of the insertion or deletion, but you also have an additional penalty here for starting an insertion and deletion. And that's because insertions and deletions are much less common in comparison to substitution mutations. But once you do have an insertion or deletion, then having one that's a little bit longer is not that much less likely. And so you assign penalties for two bases matching one another, and then for two bases not matching one another, which would be a substitution. And then you have a penalty for starting a gap of an insertion or a deletion, and then a different penalty for extending a gap for an insertion or deletion. And this will help allow you to quantify the two different representations. So here you would have two mismatch penalties for the two different substitutions, and then a penalty for opening and extending an insertion of one base. Whereas with this representation, you would open two separate insertions and deletions, and then you add the extension penalties to reflect the length of those insertions and deletions. And so if you assign values to substitutions, gap opening and gap extension, here we choose just any penalty for substitution first, and then we want a gap to be less likely than a substitution, so it has a higher value, but then it's not that much of a penalty to increase the length of that. Then once we have those numerical values, we can calculate whether one representation versus the other representation makes more sense.
Hey Tim,
Yep.
there is a question, and I think I'm gonna allow Changchen to speak, so Changchen, are you able to?
Yeah, so for option 2, your deletion and insertion is actually connect to each other. So I think that it's just one gap opening plus 8 extensions. When you count it as 2 gap openings.
Yeah, so it's being counted as two gap openings because it's not two deletions or two insertions that are adjacent to one another. It's two separate variants, where the first one is a deletion, and then the second one is an insertion. And so you have to penalize for opening the deletion and then for opening the insertion. Does that make sense?
Okay. So my second question is why match is 0? So ideally you should give some bonus for fully matched alignment.
Yeah, so there's prior work that has shown that you could have a bonus for matching variance, but that's actually equivalent. Basically, you can just scale and shift everything so that the match bonus is 0. And so I've just chosen m equals 0 to start with, just because it simplifies things and then you don't have to worry about that. Yeah, is that what you were gonna ask as well? Okay.
Yeah.
Yep.
So for the option 2, for example, for the first deletion, the formula is 0 O plus 3E. So my question is, we have three space in the deleted, right? And it has been reflected in the part of 3E. But besides that, we have to add the opening. My point is here, seems like for the first space, we have double punish, right? Because
Yeah, so for the people over Zoom, if you couldn't hear that, the question is if the kind of the first space is almost double penalized, because over here we can see if you have just one base, then you need both an opening and an extension score. And again, for that, basically what you could do is set your opening score to O minus E. And then, so basically, yeah, you can just kind of, as long as you choose your opening penalty to be the right value, then you could formulate it either way, basically. Yeah. Okay. So moving on, this slide has a lot going on, but basically the whole point is to show that there is kind of a whole range of values that you could choose for your parameters, gap opening, extension, and substitution. So here on the x-axis, we just have the penalty for starting a gap relative to the cost of a substitution. On the y-axis, we have the cost of extending a gap relative to a substitution. And then kind of as you move through this space, you can choose any design point. And as you get closer to the origin, your alignment will start preferring insertions and deletions. As you move away from the origin, you will start to prefer substitutions. As you move towards this line, you will start preferring to have separate gaps. And in the extreme case, if you edit distance alignment where you don't have any gap opening penalty, then you can have as many separate gaps with no additional penalty. And then along this axis, you would prefer to merge your gaps as much as possible. And so based on the application, you'll choose a different design point in this space. And so short and long read aligners are typically clustered around here, which we've labeled design point C. Assembly-based aligners, they tend to prefer merging gaps as much as possible. So they'll end up closer to y equals 0. And then structural variant and copy number variant aligners typically want to allow insertions and deletions as much as possible, especially in like repetitive regions where they are more likely than substitutions. So those types of aligners will be clustered down here near the origin. And so moving forward, I just kind of would like to highlight, I have like design points A, B, C, and D kind of across the space, just to kind of show different representations that could be used in reporting variance. Yep.
I assume this mismatch penalty is just kind of dependent on O and E, right? So that's why it's here.
Yeah. So everything is plotted relative to the substitution penalty. So basically, once you normalize everything so that the match penalty is 0, and then you divide by the substitution penalty, then you've kind of constrained it to like a 2D space.
Yeah.
Where then you can exploit the different parameters. Yeah. Okay. And then now I'm going to talk a little bit about what doing this changed for our evaluations. So the first example is we had a tandem repeat benchmark that had an original representation that was pretty far out there. So if you go back to this slide, I believe it was somewhere up here, and that it very heavily preferred substitutions. So what we did once we normalized it to point C, which is much more typical of short and long read aligners, we found that the number of SNPs almost halved, and that there's significantly more indels now. So these parameters are very important in the representation, especially in complex or repetitive areas of the genome, where there is more likely to be a combination of insertions and deletions. And because you have these long repetitive elements, those insertions or deletions could happen anywhere along a pretty large range. And then it becomes more likely that they interfere with other SNPs and variants. So for this data set, we found there was a huge impact. And here's just an example where the initial representation used 12 SNPs and one very large insertion to represent some variation. Whereas once we normalized it to design point C in the middle, which is more typical, we found that this set of changes is actually exactly equivalent to these two insertions, which is very interesting and not something that you can really notice very easily just by inspection. So some other contributions that we made in this work were efficient variant clustering, so that you only have to deal with the representations of variants that are nearby and could actually impact one another. And then compared to prior work, which does these evaluations, we also allowed inexact variant matches during the benchmarking and variant comparison. And then we also changed things so that we used phasing information for the evaluation, because the benchmark that we compared against was designed for short reads where phasing information was not available. So we extended it to work with phasing information. And then we also had proposed some distance-based evaluation metrics. So I'm not going to cover most of these changes. I just did want to briefly show an example of inexact variant matching. So here's an example kind of similar to the previous one, where this one large variant plus another substitution was very similar to this other set of variants in our ground truth. But in this case, it's not exactly the same. I think there was like one base different, which is why the match is around 98% instead of perfect. So this is an example of where now our tool is able to figure out that these variants are actually pretty much equivalent to one another and appropriately assign credit during benchmarking. And then in order to evaluate, we looked at data from Precision FDA's Truth Challenge. So they had 64 different submissions for the same individual using a whole host of different sequencing technologies. And so this was the perfect data set for us to look at. And then just a figure kind of showing the high level results. So we changed the variant representations to the original representation in black, and then the 4 different design points that I pointed out earlier across the space, and then looked at the F1 quality score, which is a balanced metric combining precision and recall. And ideally for the evaluation, you don't want your F1 score to change at all depending on the variant representations. So ideally you would just have a perfect line. And we can show that our work gets a lot closer to this than the previous work. Okay. The next thing that I looked at, which is a paper that should be coming out on bioRxiv, like either today or tomorrow, was actually kind of the overall methodology for evaluating small and structural variants. So right now, people evaluate them both separately. So my first work was focused on just small germline variant calling evaluation. So just SNPs and indels, and not looking at structural variants, which are much larger. And if we look at this example here, there is an issue with evaluating them both separately, because now that we have intelligent variant comparison, we will find cases where a large structural variant is actually equivalent to multiple smaller variants. So if we were to separate them into 2 different files beforehand and then do the evaluation, we would find that our query had this large structural variant, but our truth had no corresponding large structural variant. So it would think that's a false positive. And then if you look at just the small variants, it would actually also consider this a false positive because there's nothing at the same position. And then it would consider all 6 of these to be false negatives. So we need something that can work with both the large and the small variants at once.
There's another question online from Cheng Shen again. Which design point should we choose in practice? Should we just use OEX combination that minimize the number of variants?
Yeah, basically we did a lot of thinking about what if we could propose kind of like a universal like recommendation. But honestly, at the end of the day, it really does depend on your application. So we have design point C kind of in the middle of what long and short read aligners usually do. And that's kind of like what we settled on as reasonable for kind of like non-super repetitive areas of the genome, where just like genes, where there's like a normal amount of variation in the sequence. However, like we saw that the structural variant and copy number variant aligners typically choose very wildly different parameters. And so if you're trying to identify that type of variation, then you probably should choose different parameters. So the most important thing is that you're aware of what effect that parameter choice has on both your query and truth data set, and that you choose the right one for your application, and that across different data sets you're working with, if you're comparing them, you should make sure that you're using the same representations across those data sets. Yeah.
Can I follow up on that question?
Yeah.
So presumably there's biological processes that are leading to SNPs, these indels or structural variations. So even if you end up with the identical reference and sort of query one that is true, and you don't have a way of deciding which just by staring at it, there is a biological answer to how that arose.
Yeah.
through homologous recombination errors or whatever. So
Mhm.
just to throw up your hands and say their design space is open to your own interpretation, it seems a little bit like you're giving up when there might be an answer that may not be accessible, but there is an answer
Yeah,
here.
that is a good point, that like there was some type of biological thing that happened that ended up with that new sequence.
Right,
But the problem is a lot of the time, you just have that new sequence in the old sequence,
yes,
and you can't really go back and figure out like what set of variations led you to that point. I guess in some cases it might be possible, where like you have history of other patients that only had like one mutation, and then you have yours which has several, and then it's most likely that it contains that previous mutation plus something else. So there, it would be possible, you're right, in some cases to using external data, kind of try and go back and resolve and figure out what makes the most sense. But I guess without doing that and just operating on the sequence information that you have, then you kind of just need to choose parameters that reflect the relative likelihoods of those events happening and kind of work with that, even though you're right, it's not perfect. Yeah, does that make sense?
Yeah.
Okay. So the second project is basically on evaluating both small and structural variants together. So historically, they've been evaluated entirely separately. And the main reason for this is that short read sequencing was and still is kind of the dominant whole genome sequencing approach. And so they would use short read data to make the small variant calls. And then prior work, vcfeval is designed for just small variant comparison, and it requires the exact matches. Whereas then if you wanted to do structural variant calls, more recently, long reads came onto the scene. And even though they were far less accurate and not really good enough for the small variant calls, because they're so much longer, they will still map correctly. And they were used to call structural variants. And then tools such as TrueVary, which use kind of heuristics in terms of like percent match, the size of the variant, how nearby they're located, would then be used to evaluate whether or not those structural variant calls were correct. But more recently, we've seen the emergence of pipelines that can call both small and structural variants from one sequencing workflow. And typically it uses long reads as the backbone. That can be polished with short reads, or it can be supplemented with like Hi-C or optical genome mapping data. But basically now it is possible to call small and structural variants from the same pipeline. But at the same time, the current methodology for the evaluation is still to do small variants and structural variants separately. And even this delineation between small and structural variants, right now the threshold is at 50 base pairs. But the reason that 50 base pairs was chosen doesn't really have any biological origin. It was really chosen because short reads are generally 100 bases. Once you start dealing with insertions and deletions longer than 50 bases, then the short reads kind of struggle to map correctly. And basically the precision and recall of structural variant calling falls dramatically once you start looking at bases longer than, variants longer than 50 bases. So the solution that we worked on was a joint evaluation of both the small and structural variants at once. And so what we did is we extended vcfdist, so instead of just working with small variants, it can also do structural, and that required a bunch of like performance and efficiency improvements in order to be able to handle those larger variants. And then I'm just going to show kind of some of the results that we ended up finding. So this also has a lot going on, but basically the top is showing the false negative rate and the bottom showing the measured false positive rate. It's on a log scale, so everywhere from 0.1 to 100% error rate. And then we looked at 3 different whole genome data sets, Hi-Fi ASM dip call, Q100 PAV, Hi-Fi ASM from Genome in a Bottle. And then for each VCF, for each set of whole genome variant calls, we looked at 6 different subsets. So we looked at SNPs individually, indels individually, structural variants individually, small variants, which is the SNPs and the indels, what we called large variants, which is the indels and the structural, and then all variants. So the only 3 categories that are really typically used are the small variants, the structural variants, and then our pipeline uses all variants. So what you want to look at is for the SNPs and indels, the performance improvement from moving from just small variants to working with all variants. And we can see that for all 3 data sets, for both false negatives and false positives, we can see that there's a reduction from the orange bar to the green bar, which is the move from small variants to all variants. And then for the structural variants, the move from red, which is just structural variants, to when you look at structural insertion, deletion, and SNP, and we can see an even bigger performance there. So everything's on a log scale, so it's kind of hard to see what's going on. But we found that there was an average around 25% decrease in measured false negatives and false positives for SNPs and indels, and around 50% decrease for structural variants. And again, that's going back to the cases where there's several smaller variants that match up with a large variant, and vice versa. So the next thing we did was we compared to prior work. So vcfdist is our tool that was designed for small and large. That's in green. In red is vcfeval, which was just designed for small variants. However, they do go up to structural variants up to 1,000 bases. So we were able to get bars for that. However, because they require exact matches, you can see that there's a bigger and bigger difference between that prior work and our tool as the variant size increases, because it's less and less likely that you're going to have a perfectly exact match. And then TrueVary works with indels and structural variants. It doesn't evaluate SNPs at all. So we have 100% false negative rate and 0% false positive rate for that. So that's kind of the high level accuracy results. And then something else that we were able to do is evaluate the phasing on our 3 different data sets. And what we noticed is that our results much more closely matched our expectations than what WhatsApp does. I guess I forgot to kind of put in explanatory slides, but phasing is basically when you have variants that occur on only one of the 2 copies of your genome, and you want to say that it happened on the paternal copy or the maternal copy correctly. And so as you have a bunch of variant calls and you're saying, okay, this one was on the paternal, this was on the paternal, this was on the maternal, you want to get all of those correctly. And there's 2 types of errors that happen. A flip error is when you accidentally place one single variant on the wrong haplotype. And then a switch error is where basically that variant and all following variants for a while accidentally get placed on the wrong haplotype. And the reason this happens is if you are doing your sequencing and you know there's a few variants over here and a few over here, and you don't have any reads that span from one set of variants to the other, then you can't link the 2 together effectively and say whether or not they happened on the same copy. And so basically we can use this variant comparison tool to then tell whether or not the variants were on the correct haplotype or not. And we compare to existing work WhatsApp. And we see that, so this data set originated from a hi-fi ASM de novo long read assembly. And this third data set originated from the same de novo assembly, but just using a different method for calling the variants. So we would expect very similar performance in terms of switch and flip errors between the 2. We see that WhatsApp finds 610 switches and we find 494. But then on the second data set, we again find exactly the same number of switch errors. But here, the number of errors that WhatsApp finds has nearly doubled. And we actually find that this is due to the variant representations, which I'll explain on the next slide. And then in contrast, this second data set was generated from a Q100, a very high quality de novo genome assembly. And we found that there were only 6 switch errors, which is more similar to what we would expect than WhatsApp, which still finds over 300. So there's a bunch of noise originating from WhatsApp. And now I'm going to explain why we found that was the case. So this slide right here, if for now you only look at the 2 variants in red, so at chromosome 1, at this position, there is a deletion of TG, and in this format, the genotype field is GT, which is what we care about. So this 0, 1 indicates whether that variant occurred on the first copy, the second copy, or both. And we see that in our query, we said that the variant occurred on the second copy, whereas in the truth data set, we say that it occurred on the first copy. And so in isolation, these 2 are the exact same variant at the exact same position. We place it on one copy, the truth places it on another copy. So that is most likely a flip error where we accidentally assigned the variant to the wrong copy and we made a mistake. And that's what the existing tool, WhatsApp, will tell you. It will report, yes, this was a mistake. However, if you take in context all the variants around it, and it is kind of a mess, but vcfdist is able to, as its benchmarking decision, decide that all of these calls are true positives, because once you account for all of the variants together, the sequence between your truth and query for both haplotypes matches exactly. So even though if you consider just the individual variant representations, and it really looks like there's a flip error here, once you take into account all the surrounding variants, it's actually just the variant representations that differ. And the 2 sequences that they report are exactly the same. So here we can see the real difference is, here the truth says a T and G are missing. And that basically, it just places the T later on instead of at this same position. So it's only the variant representation that's kind of causing all these false positives. And for that reason, going back to this slide, you can see that in terms of flip and switch errors, the existing tool WhatsApp always finds a bunch more than our tool does. And if you were to look at the variant representation chosen by this one data set, this has the most dissimilar representation, which is why it has so many more reported flip and switch errors, whereas this data set uses the same representation. And so it has 400 fewer reported switch errors.
Question online from Ray Shang, who asked, does vcfdist do phasing itself or just evaluates against the truth? How was the query phasing created in this example?
Yeah, so vcfdist assumes that the input variants are already phased. But importantly, it only assumes that local phasing is correct and not global phasing. So I briefly touched on earlier that we have an algorithm to cluster variants together. So basically any variants that are either very close to each other on the genome, or they could be separated by a long repetitive region. But because that region is repetitive, the representations of those variants could depend on one another. So we group those together into a cluster. And we assume that within a cluster, all those variants are phased correctly relative to one another. But in between clusters, we allow arbitrary phase switches. So we'll evaluate the phasing of this cluster and then this cluster and then this cluster, and then do a dynamic programming algorithm to try and minimize the number of flips and switches between the clusters. And so that way it can both discover kind of the general phasing errors, while at the same time, it doesn't do the phasing itself and requires that you have at least local phasing information in the input. Okay, well, that's actually it for my talk. The code is on GitHub if you want to check it out. And I'd be happy to answer any questions at this time.
For your talk, I do wonder whether your pattern will be also applicable to those repeats in the genome, especially like in humans, approximately like 50% of the genomes are like comprising of repeated sequences.
Yeah, so yeah, that's part of actually the motivation for this was the fact that previously the benchmarking evaluations were kind of restricted to the non-repetitive regions. And so you kind of ran into this representation issue a lot less because everything was represented very similarly. But once you start having stuff in repetitive regions and in the normal regions, then the comparison gets a lot messier. So I would say this tool can still be used for like benchmarking the correctness in those repetitive regions. However, I wouldn't necessarily recommend like normalizing the variant representation in those regions using the default parameters in this tool because like changes in the copy number of those like short repeated sequences are much more expected to happen in those regions. But yeah, it can still be used for evaluation and benchmarking in more complex repetitive regions. Yeah, the main limitation that we do have to have right now is it doesn't really do anything much larger than 10 kilobases, just due to memory requirements at the moment. Okay.
Any other questions? All right, thank you, Tim.
Thanks for having me.